Good afternoon, everyone. I'm Ed Godfrey. For those of you who don't know me, I'm a radiologist here with an interest in GI and endoscopy, and I do a lot of biopsies. And one of the lovely things about working in Cambridge is being able to interface with very clever colleagues who work in adjacent specialties, like in haemato and histopathology. And so today we're very lucky to have two speakers, the first of which is Dr. Jim Watkins from histopathology to talk to us about the sorts of things that we're able to do with the biopsy specimens and the sorts of things that are fairly specific to Cambridge, and that we have the expertise and skills in the histopathology and haematopathology teams to do quite exciting work with the biopsies that we take. So I'm just going to hand over to you, Jim, and thanks very much for coming to speak.
Yeah, so as I've mentioned, I was trained as a histopathologist, but I now do over half of my clinical work for the Genomics Laboratory Hub, which is based over in the treatment centre, and we basically do the molecular analysis for the whole eastern region, which goes up to Nottingham and Leicester. I've just been asked to come and speak today about the work we're doing on whole genome sequencing, and in particular the area that I work in, which is in solid tumour whole genome sequencing.
Apologies if the left screen isn't working, so if you can't see it, it's recording, you might want to come over the side.
Is Alice nearby? We might need some technical help. We've got a pointer, haven't we?
Are you on the Mac?
Well, it looks like on the Mac, but
This is correct. If you just click right.
yeah, great.
There you go.
I think it's all good.
Yeah, I'm in business. Yeah, so just to give you some background to it, really. So when we talk about doing molecular testing for oncology, there are two main questions that you're asking. So the first one is accurate tumor classification, and then the other one is, in a tumor agnostic way, are there any personalized treatment targets? So if you take the first one, you know, old school classification of tumors as I was trained when kind of 20 years ago, it's based purely on sort of histology and the appearances, and that does have quite a lot of clinical utility, actually. But when in recent years people have really started to drill down into the nature of these tumors, you see that within that there are actually several subgroups that have differing prognostic prospects, and also a lot of them have different defining molecular abnormalities, including treatment susceptibilities. And I've got breast cancer here, but you could do it for anything: renal, obviously haematopathology, soft tissue. This stuff is completely exploding. I was just thinking when I was writing this talk, even me as just an NHS consultant, I've described 3 new tumours in the last 18 months. So you can imagine, when you look at, you know, high throughput groups, this stuff is just exponentially exploding. And in line with that, there's also an exponential explosion of new drugs coming online. But this is kind of quite old now. This is like a timeline of approval of various different types of drugs, and they basically target molecular abnormalities, be that point mutations in genes, or in other cases in things like homologous recombination deficiency, sort of a general sort of DNA scar, as it were, signature that you can target. So, and these are important treatment targets irrespective of the diagnosis. So for those two reasons, molecular testing is kind of booming. I just wanted to talk a little bit about the general different approaches, and why WGS is different, and why we ought to bother doing it. So your standard treatment approach to making molecular diagnosis, to do what they call like kind of targeted sequencing. So this is like a panel of genes. Our panels got to about, it says sort of 200 there. Some of ours got to like 1,000 or sort of 500 sort of genes. The advantage of this, you can do it on paraffin. It's pretty quick, it's pretty cheap. And if you're just interested in a very narrow range of genes, it's pretty good for that. But then the next level up is more whole genome type sequencing. So some people do whole exome sequencing. So that's just literally the coding areas of the genome. That's another level up of coverage, but whole genome sequencing is everything. So not just the exomes, the intronic zones, the whole lot. And the advantage of that is that essentially it's a complete method, so you can detect all the different types of variants, not just in the coding areas, but in the non-coding areas, where you get very good detection of what we call structural variants. So that's things like complex inversions, dupes, and kind of large-scale deletions. And there's no other way to do this other than genome. So it is the gold standard of genomic testing. And so you may recall people talking about the 100,000 genome project. So again, sort of back in the day, where the aim was to sort of sequence 100,000 genomes, germline and somatic. But when these came out as pilot studies, I think because of local expertise, we jumped on the opportunity to put our clinical cases through it. So we've got a few different areas that we were able to sort of produce our data. The first one, the biggest series, was whole genome sequencing in the pediatric group. This is a bit of a busy diagram, but essentially the take-home message is that in about a third of cases, you'll find something unique. So that might be a germline mutation, because you do them when you do whole genome sequencing, you have a matched germline as well. It might be a drug target, might be a novel variant that's altered the diagnosis. Sometimes it kind of refines you into a particular prognostic route, which might alter the treatment approach you're taking. You know, it might upgrade you in neuroblastoma, for instance. And then we also took the opportunity to do it in sarcoma. And it was a kind of similar story. So again, in a roughly a third of cases, we'd find a treatment target or we'd alter the diagnosis. The advantage of WGS is that it can look for variants in genes that you've never heard of before. The panels are obviously selected because they're known to be involved in cancer. But if you're looking at for novel alterations, and you often are in things like weird sarcomas or weird PED things, WGS can give you the answers. And within these two studies, we did find novel entities. The third group that we've been looking at are the ovarian carcinomas. This is like a pilot study. And that they're mainly high-grade serous, but we do have a range of other subtypes as well that we sequenced. And you won't be surprised to hear that, again, roughly a third of these cases did yield an alteration that altered management. So in a lot of these cases, it was so with HRD signatures, you can use PARP inhibitors, and the best way to detect that is in whole genome, and they often don't have. There's like a family of homologous recombination genes, like BRCA, that are typically mutated in these HRD cases, but a lot of them don't have those mutations, and you only see the signature, but that still confers the response. And WGS was really good at picking those up. So this is Ross Brenton and Dr. Ifyingana, they're leading on that, and that they'll publish their work shortly. So just to talk a little bit about the indication. So as we've kind of said, the groups that we sequence at CUH is a little bit broader than that in the National Genomic Test Directory, which is the sort of nationally commissioned sort of indications for genomic testing, which get a little bit sort of political and are a bit of a football, and they move from place to place. But at CUH at the moment, we basically say that eligible patients are everybody under 25, irrespective of tumour, a SARC. known or suspected, but also CUP and high-grade ovarian carcinomas. And they've also give you this kind of get out of jail free card of any patient on which there's a clear clinical question or expect utility to WGS. And most of the patients in this group tend to be people who've had molecular testing on panels and not found anything. There's obviously something there. So they benefit from a genome. And the other group are the patients you'll be familiar with, often very young, very fit patients who failed on standard treatment, and we're looking for targets. So one important thing to bear in mind, particularly from the position of your specialty, is that we need fresh tissue. Why is that? It's often just kind of thrown out there, you need to take fresh tissue, but there's a good reason for it. So in the 100,000 genome project, initially they were happy to take paraffin, but this, you can see on the right here, this is basically the coverage, which means the number of reads at any particular locus. You can see how wobbly it is. Whereas on the left, it's nice and flat. That just shows you that that's the degradation essentially of the DNA. And if you look at this figure here, basically the grey are the number of variants that are detected in the fresh and the paraffin, and the gold are the ones that are only seen in the fresh, and then the blue are the bonus ones that aren't real, that are only seen in the paraffin. And the bottom line is that if you want to do clinical WGS, you need fresh tissue. So just a very quick rundown of the pathway as it stands at the moment. So essentially the first thing that occurs, identify the patient, often MDT, but sometimes it's kind of an acute situation. The clinician will kind of recognize the situation and do that before that step. It's very important that they have a separate consent mainly because of the germline consequences of the testing. And then we need to take a germline blood sample and then sample the tumor, which is where you guys come in. After that, we will assess it in the lab. I'll talk a little bit about how we do that. And then it gets sent away for sequencing at Illumina. We do a particular type of sequencing called kind of short read, shotgun type sequencing. It's very, very bioinformatically intensive, and there are various different algorithms that we use to look at the different types of variants. So SNV is a single nucleotide variant, CNV is copy number changes, gains or losses, indels, insertion deletions, and SV structural variants. This all gets returned to us in a data file, and we kind of chew over it. And then this is quite an intensive process as well of trying to curate which ones we think are real, because you can imagine in a genome you get a huge amount of data. But then when we've decided what's real and what the implications are, we present this at our genomic tumor advisory board and sometimes an MDT as well. So we drafted some radiology guidelines when we started to do this with Ed's help. And they are available on the GLH website. They're a kind of work in progress, so we're updating them as required, and there'll be another update soon. So one of the important components of the pathway for use is that the consent needs to be in place. And it's very important that the clinician who's asking you to do this has actually done this before. They expect you to go ahead and do that, and we want this to be available to you to view in Epic before you go ahead and do the procedure. With respect to requesting, I think Ed's going to talk a little bit more about this, but from our perspective, it's really important that it's very obviously marked for WGS, because it's kind of new for us as well. And what we don't want is for somebody to just come and like go through all this and then just check it informally, which has unfortunately happened once or twice when we first started. And we've got some smart phrases that will kind of help with that. The tissue requirements are general guidance is that for 16 gauge cores, it's 40 millimeters length, 18 gauge, it's 80. And it's important to realize that this is in addition to what you would take from to the for a routine diagnosis if you weren't doing WGS, because we can't really compromise the diagnosis. We can't split what we've got. Unfortunately, WGS is quite tissue hungry. We are doing some work with Ed to look at the yield from our biopsies, and hopefully we might be able to bring that down a bit for you. But we do need some data to before we can do that. As I say, it needs to be sent unfixed, so that the best is for it to just go fresh. And obviously, in office hours, we can do that. Just send it promptly to a lab, and then we'll kind of allocate them to frozen and fixation. But obviously, if this happens at weekend as an emergency, it is often the case. Sometimes, you know, you get these mediastinal masses and things like that. Or if somebody's been added on late onto your list and the lab's not open, there is this option of putting it in a fixative RNA later. I think I can talk a little bit. I'll give you some more guidance about this when we actually go live with it. I'm thinking it might be ready in maybe June. It's kind of caught up in, I'm sure you have it in radiology as well, the sort of quality and change committee hell kind of, but I think it'll probably be ready in June. And the significance of that is if you're going to go for that, we would have to ask you guys to allocate them to fresh and fixed. This is just a figure that Livia and Patricia drew up. It's very helpful, just essentially emphasizing the two pathways that we have going forward of within sort of working hours and outside working hours. The way we handle it in histopath, or the way that you handle it if you're doing it out of hours, essentially the rule is that sampling for conventional histopath always comes first, because this is WGS is kind of an adjunct really if we're doing a combined procedure. And again, it's just the same rules as we had before. So generally speaking, if it's a combined procedure, I'd like to get at least 2 good cores informally, and then the remainder can go into to be frozen or put into RNA later as appropriate. If it's just been WGS, it's easy. Just either fix it all, put it on RNA later, or freeze it all. Just to give you a rough idea of what we do when we get it, we basically cut sections and have a look at it to see what the tissue looks like. And what we're really wanting to do is get a sufficient number of tumor cells to be analysed, and what we like to get roughly 30% tumor cell purity. That's, you can get results with less than that, but the lower it gets, the harder it is to see the mutations from the background mutations, right? So you get all different kinds. So some of them are very cellular, like this one in the middle, but then some of them are very hypocellular or myxoid, particularly in things like sarcoma. And then when it's ready, we send the consent, the record of discussion, the request form, bloods, and the tissue over to the GLH for sequencing. So that's essentially the pathway. I just wanted to present a couple of cases where radiological biopsies were taken by yourselves that had quite a big impact on management. So this was the first case. So this was a 71-year-old gentleman. He presented with this big mass in his adductor compartment, and he also had multiple soft tissue and lung mets. And he previously had a history of pleomorphic dermal sarcoma. 3 years previous, it was only small, it was only 1 centimeter, and they can metastasize these occasionally, but they're usually just local nodal change. It's not that aggressive a disease. So this was very odd, and they were thinking, well, you know, this is kind of nearly 10 centimeters. It's probably another primary. So they were setting him up to treat him as a primary sarcoma. but we did some WGS. It looked, you know, it would do for either really on histology. And this is what we got. So let's take you through this briefly. This is a kind of circus plot. Well, you can use it to represent any kind of big data, but it's particularly useful for genomes. And essentially, each of those kind of cheese slices, as it were, is one of the chromosomes. And the blue track on the outside, that's the coverage. So it's the number of reads at each locus. And then when you come back and normalize that, you can infer a copy number from that. Then the green layer, that's the number of single nucleotide that's the number of indels per megabase. But that red one there, you see huge blocks. That's the number of single nucleotide variants per megabase. That's super high. And what we can do is then do signature analysis. So this is the sort of trinucleotide plot here in the top left, in the top right. So what you've basically got there is lots and lots of CC to TT transitions. So that's a UV signature. So this tumor is being exposed to a lot of UV light. And in addition, when we looked at the small variants at the top here, this is you get your chromosome number on the bottom. Essentially, it's a karyotype. And if you look at the copy number changes, and then the mutational profile here, and then compare it to the literature on pleomorphic dermal sarcomas, you can see that there's a very tight overlap between those two things. So when you take that UV signature, you know, it's likely to be a skin tumor. You don't get UV on the adductor compartment of your thigh, obviously. And we found out that actually this was that completely changed his treatment. You know, they would have been potentially going over and even talking about doing you know, amputations or radical limb sparing surgery. So this made a huge difference for him. And there would I don't think there'd be any other way we'd ever be able to pick this up, you know, in days gone by. And the other case that we had was a very interesting one. So again, it was a gentleman around 76. He had a massive abdominal mass, and radiologically, it was felt it's probably a sarcoma, you know, big bulky mass. I think this is one that you did, Ed. And he had a biopsy, and it kind of looked, it was cytokeratin positive, which is a kind of an epithelial immunomarker. And at that point in time, he got referred to carcinoma of unknown primary service, which is, as you know, it's a very general treatment. The survival is about 6 months, unfortunately. But because we had the fresh tissue, we did WGS, and this is what we found. So again, this is the copy number plot. So I guess this is the, you know, this is the kind of baseline. Yes, so this would be 2 copies, one maternal, one paternal. You can see most of it is green, and what that means is loss of heterozygosity. So that means you've lost one of your maternal or paternal alleles, and the others reduplicated. This is called, we call it sort of genomic haploidization. It's very rare. This you only see it in. I've only seen, you can see it in things like germ cell tumors where it's meiotic non-disjunction. And there's a couple of other tumor types, inflammatory rhabdomyoblastic tumor, things like that. But this is an unusual pattern. And then within that, you've got gains of 5 and 7 as well. Oh, sorry, this is a bit of a, I don't know what this is doing here. This just shows that sometimes we have to dissect the block to get the tissue. Yeah, and we also found some mutations here. P53 is very common. You see that in tons of different tumor types. NF2, on the other hand, is a little bit more specific. And the exact variant that we had, when you then look in the databases on Cosmic, you can find that both of those were in mesothelioma. Hadn't been reported in anything else at that juncture. And then if I went to the literature, and essentially there is a subclass of mesothelioma that has genomic near haploidization, gain of 5 and 7, and NF2. So in this particular case, we called it a peritoneal mesothelioma. So he was inoperable. But because it's mesothelioma, they're eligible for nilotinib. So he got put onto that. He had a very, very radical response, and I think they then re-referred him for surgery. So for him, his diagnosis and his treatment changed completely. And it would have been very hard to get this on a panel. You need that full picture to really, I think, confidently make this diagnosis, because the ploidy was so important. Yeah, so WGS is very much sort of the future. There's lots of additional things that we can do in the future. A lot of it's sort of quite bioinformatically intensive, but all this stuff is red hot at the moment. Lots of new signature analyses coming up that will help with diagnoses, AI classification of the data, and assigning primary site and so on, and some changes in the technology that gives even more information. So yeah, I'm a massive advocate for WGS. I'm very grateful for Ed and other colleagues that have been so great in introducing it. And yeah, so essentially, thank you, and keep doing what you're doing, and we're here to support you as much as we can. There's a couple of contacts on there as well, if you do have anything you need to ask about.
Jim, thank you for a fantastic talk, really sort of highlighting the impacts that together we can make for patients, and also amazing that you could put it in terms that even I can understand with the genomics. So I hope it won't embarrass Jim and Olivia too much for me to say that they're both leading in this area nationally and internationally. Really, this is the absolute cutting edge of pathology and personalized medicine, and it's really having a big impact for patients. So if it's okay, I'll leave a more general discussion for after Dr. Russo Barnett's talk. The next colleague I'd like to introduce is Dr. Olivia Russo Barnett, who is a consultant hematopathologist and works with my wife, and so is already someone who I have to do exactly what they say, but even more so because I know that the impact of what you tell us to do is very big for patients. So I'll hand over to you now, Dr. Russo Barnett.
Well, the impact is huge. We're already looking forward to all of it. So may I just start with everything that Jim said is true for lymphoma as well. Lymphoma is a hematological malignancy, but the considerations with the tissue diagnostics and FFP is the same. So whole genome sequencing currently as it stands also goes down the same pathway. There are slight complications that you will understand because we're special. So you'll get to understand why we're special and how we're complicating your life, and how our life is also complicated because of it. Everyone's going to start why their specialty is so special. I'm going to tell you why hematopathology is so special is because it is really truly just the 2 cell types. But we made our life really complex by breaking it down into 90 different subtypes. They're all slightly different from one another, and an awful lot goes into diagnostics as well. You remember Jim showed you they looked at a microscope, breast carcinoma, yes or no. Few antibodies are being used by immunohistochemistry, and then they move on to other bits, but they established the diagnosis here. We do 17 plus immunohistochemistry sections quite often, which means we're just cutting, staining, going back and forth, comparing all the time. So if you take the same amount of core biopsies or otherwise from a breast sample than you do from a lymph node, we do an awful lot more during the diagnostic histopathological workup of it. And we do molecular testing very conservatively because we try to save our sanity by reporting all the molecular on the other side. So we do require 30 to 40% of our cases to go through molecular testing to either establish the diagnosis of lymphoma, or ask you guys to take more sample because I can't quite get to it, or have results that will influence first-line therapy decisions. So molecular testing for us is incredibly important, and actually, in part, this was one of the main drivers where lymphoma diagnostics, along with myeloneoplasia diagnostic, was centralized in the early 2010s into hematological malignancy diagnostic services. That's where Ed's wife works, and she's my boss, so I have to do well here. which means that all of hematological diagnostics has been moved into centers across the country. So we kind of are histopathologists, we in the audience and myself, but we don't work in a histopathology lab, we work with the rest of other modalities that are required to establish the diagnosis. Also, as you know, more often than not, there's absolutely no follow-up excision. So the tissue you gave us is the tissue we have to use for all that complex diagnostics and the molecular and everything else that's required afterwards as well. And again, another significant causation, we have to do it really, really quickly. Now, you can imagine if you centralize something, there'll be places where that work is no longer performed, and therefore whatever they do comes first, and they use the tissue, they have a look, and then they send it to us. So by the time we receive the tissue to work up and diagnose, something's been done to it, tissue has been lost. So for us, it's always a significant consideration to preserve tissue and make the most of it, whichever way we can, and get to the diagnosis as soon as possible with the best quality of sample that we get. And also another consideration here as well, I'm not sure how much uniformity there is in practices within radiology about what gauge to use, how many courses to take. It's absolutely zero uniformity in pathology about how you handle those. You put it in one block and throw it at the same time, or take it apart in 3 blocks and preserve tissue. Zero. I can't predict what they do with the tissues, and therefore I have to do my best over here. So again, this is just a depiction of what I was telling you about. Some of the cancer types, you can establish malignant or not on a single pink and purple H&E stain section, and then you can take it from there, and you can treatment perhaps can be planned or indicated, then you can do molecular testing. Jim, you can tell me if I'm incorrect about all of that. But for lymphoma, we oftentimes need additional molecular to start treatment. And there are some very, very tricky neoplasia lymphomas where we're just pushing and pushing and feeling like spending money on molecular, we're never going to get them. We definitely need an excision or more core biopsies to do it, or we need to get to the decision as early as possible. And so this is just to show you a little bit the region that we're working in, and this is the East GLH catchment area, and just the challenges that are, but I'm going to show you our little bit, which is kind of the far east side of it. So there are some hospitals in our region where there's no pathology department. So if the radiologist takes the biopsy there, they need to pack it up and ship it off to the closest histopathology department. So the tissue already travels a little bit. Then in those histopathology departments, they handle the tissue, they cut into the block, they waste some tissue, they have a look at it, maybe do a bit more work, and they think, oh, actually, I think it's lymphoma, but we don't report lymphoma, let's send it to HODS. Then the samples come to HODS, the hematology services. In East Anglia, there are 3 of them, Leicester, Nottingham, and us. So by the time we get the sample, that's been through a number of steps. Our department is actually physically separate from histopathology, and therefore, if we need to do any work, they do it, it comes back to us, and if we need molecular, then we need to cut into the block again and send it off to molecular testing. So there's lots of cutting into and going back to the block, lots of using of the tissue as well. So again, I thought it was a very, very interesting example. If you're in Great Yarmouth, fortunate and unfortunate, there's no PET department, they take the biopsy, goes to Norwich, they do what they need to do, comes to us, we look at it, we think lymphoma, histokindly does our immunohistochemistry, comes back to us, we think, oh, hang on, we need the molecular, then they send it over to GLH, comes back to us for reporting. So we do this magical roundabout of trying to get through the patient. Then we get all the emails, we're chasing this biopsy, can you please tell us, look at the slides on your desk and get on with it. I'm sure that's a familiar sentiment to you guys as well, being chased for results. So, but what if there's a way to remove as many of the steps as possible and make everything bit better and faster for patients? So that's where we thought that we actually already had something working quite well with Papworth and chest physicians, where they were sending us additional samples, fresh samples for flow cytometry. Granted, quite a lot of those turn out to be carcinoma, but the idea was there that they're sending to us, we do flow cytometry, that's useful. And then when you actually come across some lymphomas in there, that's incredibly helpful, because what flow cytometry does is exactly the same as the immunohistochemistry does, detecting the proteins that we'll be detecting by immunohistochemistry, by doing it really fast, and also can tell you if your cells are monotypic or not, either clonal or not clonal. So that's very useful. It's significantly faster than traveling around East Anglia and then using up the tissue and running out before you can complete your workup. And also another benefit, again, is you can send a tissue to us as well directly. And now you think, yes, you got cells, you can shooting through flow cytometry, what do you do? There's a tissue. Well, Cherise can tell you that if you send us a piece of tissue, then she can disaggregate it and the cells will come apart. Again, slightly different to cancer. Cancer is a big group of all sorts of diseases, some more cellular, some less stellar, some more myxoid, some more fibrotic. So you would really be risking losing tissue, not getting enough cells or not. But lymphoma, it's mostly similarly sized round cells who like to disaggregate. So by the time you put in saline, you touch it, comes apart, you can see it actually sloughing off the surface of it. So it actually lends itself for disaggregation and flow cytometric assessment, really. And again, if you think about it, if you have a fresh tissue core that you put through the flow cytometer, do the same protein level detection that you do in histology, and in parallel, your sample somewhere in East Anglia or within CUH is going about. By the time you receive the sample, you've done all your investigation in the flow sample, and you sent it already off for the molecular testing. So it's already running. In some cases, you already get results back by the time you get your first HNA to look at and make your mind up what you want to do with it. And again, how much tissue you need for this? We don't yet know, but we're keeping an eye on it, and Cherise will tell you a little bit more about it. But to be honest with you, every single thing that you guys have sent through to us, we've been able to do all standard of care molecular testing and all the fish that is required. The standard of care is doable faster and more reliable as well. And of course, if you can already tell by flow cytometric analysis that this cell is neoplastic, you don't need to do molecular testing in the GLH that will tell you it's lymphoma or not. You don't need to do clonality. They will be happy they don't have to do the work. And again, as Jim said, fresh tissue is better quality. So actually the results we're getting back, not only faster and more reliable, but better quality as well. And if you have enough DNA, you turn out that actually, rather than doing NGS, you want to do whole genome sequencing, you go back, we can send the patient, and you can still use the same material because we have it and we already used it. Plus, because we're doing the same typing, we can tell you that, yes, it's lymphoma. Yes, it's got 40% lymphoma in it. We don't need a frozen section. You'll see how a frozen section actually is not particularly helpful for us. So I'm going to hand over to Cherise Wilton, my colleague in flow cytometry, and will tell you a little bit about what we found with our samples that we already have looked at within plots.
Hello. So this was just some provisional data of looking over the last year of the samples that we received for flow cytometry, which ones we were able to identify any malignancy in, and just compare that to the histology results. So as Livia already mentioned, if we receive a core, say it was taken at CUH, we ideally would like that as quickly as possible because it's a fresh sample. It comes in saline, and then we can disaggregate that relatively quickly. So within 1 or 2 hours, we have taken that core biopsy and we have put it over some mesh, and we have got a single cell suspension, and that is pretty fresh. And from there, we'll then add the antibodies to it, and we'll be able to phenotype it and sometimes get those results out the same day. So we wanted to know from the cases that we've done that, were we getting, were we able to identify malignancy, and also is that comparable to histology? So of the samples, there was 132 lymph nodes or tissue biopsies. So sometimes these aren't lymph nodes, as you're more aware, sometimes they were small bowel biopsies, mediastinal masses, etc. Or finally the aspirates. These ones don't actually need disaggregating. We looked at them. We could see that there were 45 of these that needed disaggregation that I've just mentioned. And then of them, 32 of them identified some disease in flow cytometry. So that's, we were able to identify that there was, say, clonal B cells in there. And there was a 93% consensus between the flow findings and the histology findings, with a couple of outliers, which could be explained.
Do you want to explain?
Yeah, you're welcome to, yeah.
So some places we found that flow cytometry was better. So if you get the DBS sample is blood stained, there you're actually sampling the blood as well as you're sampling lesion, and therefore flow cytometry picked up monoclonal B lymphocytosis in the sample that you and I couldn't see down the microscope because our eyes are not as good at, light microscopy as flow cytometry. So actually there's a bit more, it's better at picking up. And the cases where there was discrepancy, i.e. Flo didn't find the disease and they did find it by histology. They were specific to the types of Hodgkin lymphoma. We don't really see by flow cytometry, but we do the touch breaths. We actually can look at the cells, but we wouldn't expect to find any atypical findings in there. Or there are a couple of cases we actually don't know because they didn't refer them to us. So we don't know whether there was lymphoma there or not. They just didn't pick it up or were suspicious and stand it down. But I think that is a very, very good result. And just to show you just again the timeline, and this is without any optimization, because as you can imagine, we've not really got the money to put additional resources in. All of that work is being done by you guys doing your bit, and our guys are doing our bit as well, and we're pulling together for the patient's purpose. But again, if your samples are running parallel, by the time you get your H&E and or immunos out on the sample within CUH, you have your FISH results back. You can sign it out. It's not on my shelf. I'm not waiting for the GLH. No one's bothering me. It's signed out without a reasonable time frame. But you see the 22 days as where we have to do additional testing that were not thought of at the time of flow cytometry, but that can be optimized as well. So I thought I'd give you some examples. I think this is one of Ed's cases actually. CT-guided retroperitoneal mass biopsy, we had 4 million and we had fresh tissue. 4 million went to histo, fresh came to HUDs, looked at a microscope, diagnosed diffuse large B-cell lymphoma. By the time it was diagnosed as such, we already had the FISH results back. So day 7, we could sign up the case. Now, our usual turnaround time on cases within CUH that require molecular testing is 32 days. So I think that's pretty good. What do you think? So I think that's actually quite nice. And again, the point to make here really is 3/4 were taken, one with the histo, and this is why we're being problematic because histo is separate to us. They're on level 5 very soon down in Discover Drive, whereas us in HODS on level 3. So it's very, very important that the right tissue goes to the right place, because if everything goes to histo, so, oh, it's a fresh core, there's a 4 million core, does HOD need it? Do we need it? Is it double GS? Is it not double GS? There's a bit of confusion around it. But if it's clear that one is for flow cytometry and it labels as such and the order is placed for it, one for histo, even if it turns up in the wrong place, we can troubleshoot and make sure it gets to the right place straight away. But it needs to be clear what it is. Molecular testing is not quite enough of an information, and I understand how clinicians are, so sometimes that's not quite clear. Sometimes they want double GSN, GS fish, and a lot, and everything just goes to HODS, and you just have to spend some time to figure it out. This is another example. This is a 20-year-old male. He is teenage young adult, and therefore tissue was taken for whole genome sequencing. Teenage young adult, solid tissue goes to histopathology, as it should really at this time point. They took a frozen section. That's what it looked like. I don't know if someone wants to volunteer how many of those blobs there are lesional or not. I don't know, Jim, what do you think? He's not volunteering.
Glad you asked Jim and no one else.
I'm trying to be kind here, but you know, morphologists, I mean, if you can tell me how many of the cells are different to the others in that, which is, I think, it's a fair question to anybody, I'm not sure you can tell. I can't tell either, and I get paid to do this. But thankfully, we have flow cytometers. This was actually a reactive lymph node. So we nipped it in the bud. We didn't go for whole genome sequencing. We didn't spend the cash, but you know, we got to it fairly quickly as well. And this is another example, which I feel is really powerful. This is actually surgical resection, because some of the bowel people are really starting to get interested in it. The transplanters are really on it. This is a 4-year female interception emergency at resection. He had right hemicolectomy, and also, as you can imagine, the pediatric pathologists are really on top of that game as well. So they know how well you can tell lymphoma from not lymphoma for resection and send us for flow cytometry as well. I mean, here actually I'm a bit cheating because it looks like Burkitt lymphoma even on this pink and purple. So you know, chances are that that's going to be it. But you can see here within 2.5 hours we confirmed malignancy and suggested the phenotype was Burkitt lymphoma. But they forwarded a FISH report. And at that point, we still didn't have the histology formalin case to look at. Again, slight cheating because a larger piece of tissue needs slicing and fixing. But the point, I think, is well made that it is useful to have that sample. And quite frankly, if they wanted whole genome sequencing, we could have probably retained that chunk of tissue, send it off without needing to bother anyone in histology with that. So as Jim said, we have put together a visual, because I'm a morphologist, as you are as well, I assume. I like pictures and I like things to be there when I need them rather than reading through lots of pages. And I really, really would like you to send us any feedback via Ed or directly about it. It's literally just a summary of where fresh tissue is useful and how much do you need and what needs to be in place for that to go ahead and where flex should go up. So Jim has shown you the one side, whole genome sequencing, which is all of lymphoma as well currently. It's easier because that goes to one place in histology. And quite frankly, I cannot go to Nottingham, Leicester, Peterborough, Ipswich to handle their lymphomas in their histo departments. At the moment, it's got to go there. But if we build up the evidence that flow cytometric assessment is better, then there's a potential to move it that sideways as well. Of course, needs more evidence. And again, but the second part I talked about a lot more here is the standard of care diagnostic pathway for lymphoma. And I think this is where you come in as well. We don't want everyone to be fresh tissue sample because you don't have the capacity, we don't have the capacity, and quite frankly, it's not helpful for everybody. But if you think that based on radiological appearances, and again, it's easier, I think Tulip told me it's easier on a scan than it is in ultrasound when someone pops up with a lump, and I can fully appreciate it. But if you think it's more likely to be lymphoma than not, it is incredibly helpful, as we've seen, to have an additional fresh tissue core sent to us where we can expedite all the additional things. Now, as you can see it here, then there's unfortunately additional tasks which I'm sure Ed will talk to you about that needs to be split. One needs to go one way, the other needs to go the other way. I feel that there are definitely benefits to it. And you can see Patricia put together this visual. We do have Patricia tissue pathway coordinator, so you can think of anything that could be helpful in radiology for this to happen. You need someone to put on the order, fill in the form, make sure you put it in the right pot, etc. Tell us. She can help as much as you need her to help. Fill in forms, chase down anything, whatever you can think of, let us know. So I think that's all I have to say there.